welcome back. This is lecture number 11. We're going to talk about the infected cell. And you may say, well, haven't we been talking about the infected cell this whole time? Yes, but this is a different approach. We have so far focused on gene expression, genome reproduction, and assembly, right? All of these things, all of these activities depend on the host, of course, on metabolic, biosynthetic, signaling, and trafficking systems. So today, we are going to take an integrated look at the impact of virus infection on the host cell from a point of view of these kinds of activities that happen during the reproductive cycle. And we're going to look at 4 distinct areas of the infected cell. First, signal transduction, gene expression, metabolism, and remodeling of cellular organelles. A lot of this is quite new, only been worked out in the past few years. So first, signal transduction. Hopefully you all know what signal transduction is. If not, I will explain it here. Signal transduction is the mechanism by which cells sense their environment. So here's a cell. There are nutrients outside. The cell can sense that and respond appropriately by dividing, for example. In fact, signal transduction pathways govern every aspect of cell physiology and conduct, what a cell does in its particular environment, wherever that might be. And here on the left is an example of such signal transduction pathways. Typically, we have cell surface receptors that will engage ligands. And when the ligand is engaged, a cascade results. And these are green arrows in my pictures that I always indicate stimulate. So in this case, engagement of these receptors by their ligands activates the PI3 kinase, which is a very important kinase, a central kinase that will then phosphorylate a substrate. And you have a chain of phosphorylations typically, and that's what we mean by signal transduction. So in this case, we stimulate PI3 kinase, which will phosphorylate another target, which in turn will phosphorylate another target. And then when we have these red arrows, that means inhibiting. And these can get very complicated. You kind of have to work through because you have multiple inhibitors and stimulators. So for example, phosphorylation here is inhibiting AKT, but AKT is inhibiting these two, which in turn inhibit this. So that releases everything downstream of this, right? Because normally Reb stimulates those. But if you inhibit it, you release that. So eventually, in this case, we have things like cell proliferation and growth, autophagy, either being stimulated or inhibited. So if cells have growth factors outside the cell, they will produce, they will divide and make the genes that you need to divide, for example. Now, the key here is that virus infections can change signaling to promote their own reproduction. Of course, they have to do that. And this is something we've only discovered recently. So on this picture are a number of viral proteins, like adenovirus E4-ORF1, that's a protein, influenza virus NS1, etc., which interfere or stimulate. So here we have these viral proteins stimulate PI3 kinase. And probably that's so the cells divide and grow. That same stimulation will inhibit autophagy, which would be the cell killing itself, of course, digesting itself. In other cases, the viral proteins inhibit. So here, VSV is inhibiting AKT, which usually inhibits TSC, so that relieves the inhibition on TSC, and then you can follow all the downstream events. Right, so that we're going to look at some examples of this today. But at all of these pathways, viral proteins interject either in a positive or negative way to ensure that the cell environment is proper for their reproduction. So that's the end goal here, is to make sure that the cell is suitable for virus reproduction. So here are a couple of examples of how signaling via PI3 kinase, phosphoinositol 3 kinase, it phosphorylates PI3, sorry. In this case, we have 3 examples of how signaling via this kinase, protein kinase, facilitates virus entry. So here on the left, we have adenovirus binding to its two receptors and being taken up by a coated pit. Eventually gets into an endosome, right? And what's happening here is that the binding stimulates a phosphorylation cascade through these proteins and eventually PI3K. And that signals through some other proteins, including the Racaniello kinase, the RAC. And these then loosen up the actin microfilament so the vesicles can move in. Remember, the plasma membrane, just beneath the plasma membrane, there is a layer of actin microfilament. So vesicles can't just move in willy-nilly. They have to, that microfilament network has to be loosened up. And so the virus binding stimulates that. It makes perfect sense, right? And if you interfere with this pathway, the virus can't get into cells. So that's one example. Here's another one, influenza virus binding to the cell surface. Again, we're going to see the loosening up of the actin microfilament, one thing, but other things happen. And what happens for influenza virus, the binding happens in areas of the plasma membrane called lipid rafts. These lipid rafts cluster after virus binds. They activate PI3K, which helps loosen up the actin microfilament. But it also activates the vacuolar ATPase, which is a pump that pumps protons into the endosome. Remember, you need acidification of the endosome in order for the fusion to occur. So that really facilitates that. It's really amazing. And remember, the fusion of the viral membrane and the endosome membrane liberates the ribonuclear protein so it can go into the nucleus. There's also another interesting activity that's been recently discovered, and that is the virus appears to bind to this calcium channel, voltage-dependent calcium channel on the cell surface. It doesn't enter that way. But what it does is it binds and it makes calcium go in, and that somehow facilitates release of RNA from the fusion activity in the endosome. Really interesting. And if you interfere with this binding, it's very difficult for the virus to enter cells. So two different things going on there. And then finally, the last example here is Ebola virus, which binds to some surface receptor, plasma membrane receptor that's not been identified. And these are taken up by macropinocytosis, eventually get into endosomes. And remember, there's an endosomal receptor to which the viral spike binds, catalyzes fusion and release of the nuclear capsid into the cytosol. But this is facilitated by PI3 kinase again, whose signaling, again, stimulated by the binding of the virus to the receptor. It inhibits the transit of the endosome to later vesicles, which are not permissive for fusion. And so it makes sure that this step doesn't happen so the virus gets out of that endosome so it can get the RNA in the cytosol. Otherwise, in this later vesicle that this endosome would fuse with, the virus wouldn't be able to get out. So signaling takes care of that. Quite interesting. These PI3 kinases are involved in a lot of virus infections. And there's a particular relay called the PI3 kinase mTOR relay. mTOR is a master regulator of many cellular activities, including translation. And of course, viral messages need to be translated in virus-infected cells. And here we have a number of viral proteins, different viruses, hepatitis C virus, a rotavirus protein, an adenovirus protein, all binding PI3K and stimulating phosphorylation of AKT. And the herpes human cytomegalovirus protein stimulates AKT directly. And that blocks apoptosis, so the cell stays alive, increases the cellular metabolism, and increases the, it activates mTOR, which increases translation, and that's good for the virus in the end. And on this slide, another two examples. Here's hepatitis B virus X protein, and also the herpes virus 8 genome encodes a G protein coupled receptor, multipass protein, which interacts again with PI3K, stimulates AKT, and you get, again, cell division and translation increase. So there are common pathways among these different viruses. Here's a really interesting one because it's an RNA virus. And this is a viral RNA that actually blocks AKT activation to induce apoptosis. So let's go back here. So normally AKT is involved with apoptosis, right? So AKT normally blocks apoptosis. So if you inhibit AKT, you get apoptosis. These are very confusing to look at. I'm sorry about that. So here we have a viral RNA. This is a flavivirus. These are viruses with plus-stranded RNAs. They're characterized by the spike lying flat on the virus particle. And flaviviruses include dengue virus, West Nile virus, Zika virus, yellow fever virus, medically important viruses. So when the RNA is released into the cytoplasm, here's the RNA at the top. In the middle here, I've shown the RNA in kind of a simplified version. The open reading frame is shown very short. It's normally the length of the RNA. And we're showing the 5' untranslated region and the 3' untranslated region. And a fraction of the viral RNAs are degraded in the cytoplasm by this exonuclease called XRN1. This is an exonuclease we'll come back to multiple times here. It starts to chew down from the 5' end, and then it gets to the 3' UTR. It can't go any further. This structure apparently inhibits it from chewing. This doesn't happen for every RNA, obviously, or the virus would never reproduce, right? But it happens for a fraction of them. And this sfRNA, this is subgenomic fRNA, this is needed for making plaques and for causing disease in mice. Subgenomic flavivirus RNA. I'm trying to think of what the F stands for. The F is flavivirus, subgenomic flavivirus RNA. So you can make a deletion near the 3' end, which prevents the production of this sgRNA. So this mutant can't make sgRNA, and it doesn't form plaques. The wild-type virus can form plaques, but if you prevent the formation of this subgenomic RNA, the virus can't form plaques. And what this RNA does, it inactivates AKT, promotes apoptosis, so the cells break, and you can get a plaque formation. And in fact, in mice, these mutants don't cause disease. They have reduced disease. So apparently, apoptosis is important for that. Small RNA, which, is again produced in only a fraction of the genomes. Many viruses inhibit cellular gene expression. So what is cellular gene expression? Well, you start from a DNA in the nucleus, and then you have transcription to give you a pre-mRNA. Then you have polyadenylation and splicing, RNA export, and then, of course, eventually translation in the cytosol. And every step of this process is interdicted by some virus protein, and they're all listed here. So, for example, transcription by Pol2 and camping are inhibited by these viral proteins. In fact, poliovirus, which inhibits Pol2, also inhibits Pol1 and 3. So in a polio-infected cell, very quickly after infection, all cell RNA synthesis is shut off. Why? So everything's available for the virus, right? All the components, the triphosphates, and et cetera, they're all available to make viral genomes. And other viral proteins listed here interfere with polyadenylation. They interfere with splicing. They interfere with RNA export. Many interfere with translation. We'll take a look at that in a moment. And many of them actually degrade cellular mRNAs. So if any of them make it out with all these other inhibitions, then they'll be degraded. So the protein synthetic apparatus is available for the virus, so it can use it all. So that many steps in cellular gene expression are inhibited by virus infection. Here's an example of the inhibition of cellular pre-mRNA processing by viral proteins. This is a slide we've seen before, right? We have a pre-mRNA with an intron. A variety of cell proteins are binding to it to facilitate splicing. These are part of the spliceosome. And remember, the messages that are spliced then are marked by these proteins, and that those are recognized by the nuclear export system to get them exported out of the nucleus. Well, viral proteins can interfere with splicing, and many of them interfere with export. These adenovirus proteins are binding components of the export machinery. These two red ones, all the red proteins are viral proteins. VSV is binding NUP98. NUPs are nuclear pore proteins. There are a whole bunch of nuclear pore proteins, NUPs, and they're essential for export of cargo. So some viruses bind components of them. Influenza A virus, NS1, actually inhibits polyadenylation of cellular messages by binding proteins that are needed for that process. And of course, the polyadenylation of the virus is done by a different mechanism. Someone asks, is XRN a viral or host protein? It's a host protein. Poliovirus, a protease of poliovirus, 2A pro, cleaves two NUPs, 153 and 62. And in some cases, the cleavage results in unregulated export. In some cases, the export is inhibited. But in some cases, many proteins come out of the nucleus because some of these viruses are cytosolic, and they are required in the cytosol for virus reproduction. So poliovirus cleavage of these two NUPs results in a lot of otherwise nuclear proteins going out into the cytoplasm, and some of them are actually required for virus multiplication. So many, many points at which mRNA processing is inhibited. And then remember, I told you that mRNA turnover can be regulated. So messages have different half-lives. They don't all last forever. And that's controlled by sequences in the mRNA. And there are a series of cellular proteins. These are all cellular proteins here, the ones that look like Pac-Man, that are involved in degradation. So here we have a cellular message, and here we have a cellular protein, CCR4 not pan, which is chewing the poly A tail off of that message. And then once the poly A tail is gone, then this message is recognized by an exosome, which degrades the message and gets rid of it. Because if it's damaged by not having poly A, you've got to get rid of it. So that's 3 to 5 prime decay. So it's carefully regulated in the cell. And here we have 5 to 3 prime decay. The way this works is first the cap is removed. And once the cap is off, then this RNA is recognized as defective, and XRN1, that same exonuclease, comes in and chews it up. So these are mechanisms that regulate cellular messages, and of course, they will destroy viral messages, so we have to get rid of them. And in fact, many of these proteins are either removed or relocalized in virus-infected cells, so the viral messages are not degraded. So for example, this PAN protein here, which is up here, 3' taking off the poly A tail, DCP 1A over here, and XRN1. These are all degraded in poliovirus-infected cells. And again, you want to keep the viral mRNAs around. That's why certain viruses are inhibiting these. Someone asked, what is the benefit for the virus to inhibit poly A binding protein to block polyadenylation of cell messages, and then there's less competition for the translational apparatus, because those are not going to be translated, those messages without poly A. They'll probably be degraded because they don't have a poly A by the exosome. So here are some examples of how viral proteins initiate mRNA degradation. And here we have, there are a couple here, including the SARS-CoV-1, the original one, encodes a protein here called an SP1, which will chew the RNA, basically removes the cap. Human herpes virus 8 encodes a SOX protein that also chews up the cap. Herpes simplex virus will do the same thing. And vaccinia virus proteins will also remove the cap, as you can see here. And so once the cap is removed, that's a signal for XRN1 to come in and degrade the message. So again, these are viral proteins now that are starting the degradation process. And this is cellular messages that we're getting rid of. And once the cap, in this case, is removed, then XRN1, which is a cellular nuclease, comes in and degrades the message. So what's the benefit to get rid of the host mRNA? Again, to give the translational apparatus all to the viral mRNAs. There's no competition for the translational apparatus. You want to devote the entire translation machinery to the synthesis of viral proteins. That's how you can efficiently make many, many virus particles in an infected cell. First question, which of the following is a consequence of viral proteins modifying signal transduction pathways to promote viral replication? A, poliovirus inhibition of transcription by RNA Pol2, herpes simplex virus protein blocking pre-mRNA splicing, disruption of actin filaments to allow endocytosis, initiation of mRNA degradation by viral proteins. So remember, we want to know which ones are involved in signal transduction pathways. How did we do? Well, most of you got the right answer C, disruption of actin filaments to allow endocytosis, right? So that's a signaling pathway. When the viruses bind, they initiate a signaling pathway that leads to disruption of filaments. The others are not signal transduction pathways. I didn't tell you any signaling that's involved in any of these. There may be, but I didn't tell you any. We don't know of any. They're just direct effects of viral proteins. We're going to talk a little bit about translation now. And to remind you what that is, we show you the Baltimore scheme again with mRNA at the middle. Remember, all these 7 classes of viral genome make mRNA that can be translated by the host cell. So what does the host cell do with that? And this is how some translation occurs. In our cells, at least, this is how translation occurs. Of course, messenger RNAs have a cap at the 5' end, and the cap directs the ribosome to the mRNA. And it does so because the cap structure binds this protein called EIF4E. These are all viral cellular proteins here. This mess of proteins, all cellular. And then you have a series of protein interactions that bring in the 40S subunit and the initiator tRNA, the methionine, right, which is going to put down the first AUG. And then the idea is that this complex starts to scan down from the 5' end. And when it reaches the AUG, which is shown here, the initiating AUG, then you have energy hydrolysis, ATP hydrolysis. The large ribosomal subunit comes in. A lot of these proteins are released. These are all initiation proteins that are needed for this initial process. And now we have the ribosome with the tRNA met there, and it can start to read the codons and make a protein. And of course, every time there's a codon here, tRNA goes in, the right tRNA goes in, and then you make you connect the amino acid to the first one, and so on. Now, viruses inhibit cell translation. We've talked about how they inhibit cellular mRNAs, cellular mRNA production in many ways. And they also inhibit translation. So just do everything to get the whole apparatus, the translational apparatus of the cell devoted to the virus. So here's an experiment where cells are infected with poliovirus. And then at different times post-infection, we're looking at the rate of protein synthesis. And you can see by 2 hours, the protein synthesis has gone way down. And then it starts to go up again as viral protein synthesis replaces that of the cell. So we have initially an inhibition of cell translation, and it's replaced by viral translation, and eventually the cells die. So it's very quick, 5 hours. And uninfected cells continue to make protein as normal, of course. And on the right is a gel run by one of my students many years ago, where the same experiment was done, except at different times post-infection, S35 methionine was added to the medium. That's a radioactive methionine. And 15-minute, you add it, and then you scrape the cells off, extract them, and run them on a gel to separate the proteins. And you can see 0 times is a big schmear, right? These are all cell proteins. But in 1 hour, 3 hours, you can see the schmear is going away. That's a technical term, by the way, schmear. And it's going away. And eventually it's replaced by viral protein synthesis. So these other bands, which are labeled here, are viral proteins, replacing that of the host cell. And eventually the cells are dying here at 7 hours, so there's not much of any kind of protein synthesis anymore. So that's a nice illustration of shut off of host translation, replacement of schmear. I don't know the spelling. SH? Someone else must know. Yeah, give me a bagel with a schmear, right? People say that all the time. And so many viruses do this. Why? Again, they free up the translation apparatus for the host cell. So how does this work? Let's take a look at an example. And these are a variety of viruses here, how they specifically inhibit host cell translation. Remember, you can't inhibit viral translation. That would be counterproductive, right? And so here we have 3 viruses. Let's start at the top. Sorry. This is the cap binding protein, EIF4E, which will bind to the cap, and then it's connected to these other proteins, which then recruit the ribosomal subunit, as shown down here. Okay. So the translation of host cell messages works that way. And some viruses, including poliovirus and foot and mouth disease virus, they have a protease that cleaves this red protein, 4G. You can see it's cleaved right there. And so that inhibits translation because now 4E will still bind the cap, but 4G, the rest of 4G is gone, and that's what you need to bring in the ribosome. So that inhibits cell translation. All right, so that's how poliovirus and FMDV inhibit translation. But there are other mechanisms as well. In some cases, the viral proteins dephosphorylate EIF4E, so adenovirus and influenza virus infected cells. Dephosphorylation reduces the affinity of 4E for the 4G component, and it falls off. So that effectively inhibits translation. And finally, one more mechanism. There are a couple of proteins in the cell. 4EBP1 is one of them. These are 4E binding proteins, right? So they bind EIF4E and prevent it from associating with 4G. So that would inhibit translation. And so these are in the cell. These are cell proteins whose function is to keep translation down until it's needed. You don't need to be translating at full blast all the time. So in some conditions in a cell, when you need to have cell growth, take away this phosphate, which allows 4E, take away the phosphate on 4EBP1. Sorry, I'm being distracted by the schmear discussion. I'm very bad with that. Dephosphorylation of 4EBP1 allows it to bind 4E and inhibit translation. So normally the phosphate sits in there, and so 4EBP1 can't bind 4E, and it doesn't inhibit translation. Now, in the cell, there are certain conditions where this phosphate is removed to inhibit translation, but viruses can do it as well. They have enzymes that remove the phosphate, and that causes an inhibition. So 3 mechanisms by which a virus can inhibit translation. Many more, of course, but just to give you a sense, it is widespread among viruses. It's very, it's needed to get efficient reproduction. But viruses also do other things with respect to translation, actually quite extensive. We used to have a whole lecture on translation, but I had to remove it because I wanted to put some other lectures in. The cellular messenger RNAs are translated by this 5' end-dependent mechanism, which we've discussed, the cap protein. Cap binding protein 4E binds the cap and then recruits in all these other proteins and eventually the 40S subunit. And this requires all these are eukaryotic initiation factors. These are proteins like 4A and EIF3 and 4G that are needed for translation. And there are many more. There is a process on viral RNAs called internal initiation, where some of these viral RNAs are actually not capped. The poliovirus genome is not capped. The coronaviruses in general do not have caps, and other viruses lack caps as well. So how do you get the 40S subunit to the mRNA if you don't have a cap? Well, turns out there is a sequence at the 5' end of these viral RNAs called an IRIS, internal ribosome entry site. And that sequence, which is shown as the folding of this RNA here, will bind 4G directly, and that will bring in the 40S subunit. So you don't need a cap binding protein there. And this poliovirus and related viruses have this IRIS, and so they don't care if, I shouldn't say care, but it doesn't matter if 4G is cleaved to inhibit host cell translation, because that's what happens in these virus-infected cells, because the rest of 4G will still work because it can bind the IRIS and get the 40S subunit in. And then we have hepatitis C virus. What does IRIS stand for? Internal ribosome entry site. The real question is, what color is an iris? This is a joke. They come in many colors, right? Hepatitis C virus is an interesting virus with an iris where the iris actually can bind, it doesn't need EIF4G. You can bind EIF3, and then the 40S subunit comes along and binds it. This could be an ancient RNA, something just after the RNA world went to proteins. So this is what an iris looks like in poliovirus. Here's the poliovirus genome, 4,440 bases plus 1 or 200 RNAs, a poly-A tail. And you may ask, how do you know that? Well, because in the 1980s, I did the sequence of this viral genome. It was one of the first animal viral genomes, RNA virus genomes sequenced. And I probably told you already that it took me a year to do that. And 7,440 bases. And Today it would take a half hour. Anyway, at the 5' end of the genome, there's this long untranslated region. There's the initiating methionine. And then you make a long polyprotein that gets cleaved by proteases. In this sequence is the internal ribosome entry site. It actually folds. There's a lot of secondary structure there in the RNA, stem loops and so forth. And that constitutes an internal ribosome entry site. And the EIF4G can bind directly to it in the case for Picornis. And of course, in the case for Hep C, the EIF3 and the 40S can bind directly to the iris without 4G. So that's how they work. So these viruses can shut off host translation by interfering with the cap step, and they're not capped. They don't depend on a cap-dependent mechanism. These are what the folding of irises look like. There are a variety of different ones. They're called type 1, 2, 3, 4, 5, 6. You know, scientists like to categorize things, right? So we have many different types based more or less on their structures. The type 1 is the poliovirus or enterovirus iris. And you see highly structured, and then there's an AUG where the initiation starts. And so the idea is the EIF4G binds this structured RNA. In fact, on the type 2 iris, the footprint of EIF4G has been identified. A footprint means where the protein is sitting on the RNA. You can add the protein and then digest the RNA with ribonucleases, and what's left is called the footprint, and it's right there. So the 4G binds there and brings in the 40S subunit, which can then start translating at this AUG. And these are some other kinds of irises, well, different structures. There's actually no sequence in common among these irises. You can't identify an iris by looking at sequence. You have to do an assay. You have to show that it leads to internal initiation, which is typically by putting an iris in between two open reading frames and showing that the second one can be translated, which would normally not happen if there weren't an internal initiation there. There are other ways that translation is regulated. The initiation step is a big step of regulation of protein synthesis. Protein synthesis, you have initiation, elongation, and termination, right? Most of the regulation is at the initiation step, which we have described briefly. There's also some at elongation. But I want to focus on this mechanism because it turns out to be important when we talk about interferon. So here we have an mRNA with a cap, and the 40S subunit has been brought to the AUG. Now we have the met-tRNA there. Everything is ready to go. This met-tRNA is brought in as what's called a ternary complex. All that means is there are three components, the tRNA, EIF2, and GTP. When initiation begins, there's this GTP is hydrolyzed to release energy. All right, now you have GDP bound to EIF2, and it is released. And then this 40S, the big subunit will join, and it will start translating. But this GDP has to be recycled. It's in limiting amounts in the cell, so you have to replace the GDP with GTP so it can go through another cycle of initiation. And that's done by EIF2B, which is basically a guanine exchange protein. What does that mean? It exchanges GDP for GTP. So GDP EIF2 binds EIF2B, and then GTP substitutes for the GDP, and now you have released GTP EIF2, which can bind a tRNA met, make a new ternary complex, and you can do more translation. This step is highly regulated by a number of protein kinases. These are enzymes that put phosphates on other proteins, and there are three of them here. They're called PERK, GCN2, and double-stranded RNA. And these are activated in cells. These are cellular protein kinases. They're actually called EIF2 alpha kinases because they're going to phosphorylate the alpha subunit of this protein EIF2. They're activated by stress, basically. So ER stress. What is ER stress? Well, when you make a lot of viral glycoproteins in the ER, the cell gets stressed. It doesn't like that. And it responds by activating PERK, which will shut down translation. And then there's another EIF2 alpha kinase called PKR. That stands for protein kinase RNA activated. It's activated by double-stranded RNA, which is made in virus-infected cells. Typically, cells do not have double-stranded RNA in them, only when they get infected. So that's kind of a sensing for virus infection. So PKR is activated. So what happens when these kinases are activated? Well, they phosphorylate EIF2. Here you can see the GDP with EIF2 is being phosphorylated now. It will still bind EIF2B, but it doesn't leave. This thing is locked in there because of the phosphate, and you can't recycle it. And so basically you inhibit translation. You actually bind up all the EIF2B, so there's none left to be recycling any GDP EIF2 that's around. So basically phosphorylation of EIF2 inhibits initiation, and it's activated by this protein, all three of these kinases, but ER stress and double-stranded RNA are what happens in virus-infected cells. So the cell is basically trying to shut down protein synthesis because a virus is there. It has sensed that a virus is there, and it's going to shut down translation. Now, that may kill that cell, but it may protect the organism. That's the way we look at it. But that's a human-centric view. I'm not sure that's correct. But it can stop virus infection. How does this activation by double-stranded RNA work? So here is PKR. It has a catalytic domain and two double-stranded RNA binding domains. And here's double-stranded RNA. And the idea is, right, so these PKR molecules bind double-stranded RNA via this double-stranded RNA binding motif. It makes sense, right? And we believe that when you have two PKR molecules bound to the RNA, they phosphorylate each other, and that makes them active. It's now active PKR, and then it can go on to phosphorylate EIF2 alpha. So that's how double-stranded RNA activates PKR, because normally you don't want to have active PKR in the cell. It would shut down translation, and cells need to translate, right? And actually, PKR is actually regulated in uninfected cells. Here is a protein called PACT, which is a cell protein that can activate PKR in the absence of double-stranded RNA. So this is a protein that has important functions in the host cell, PKR, but it is a part of the viral defense. And as you'll see in a moment, PKR is induced by interferon. So it's part of the early innate defense against virus infection. And so here's a summary of that. PKR is induced and activated by virus infection. What does that mean? So it's induced, the protein is actually made, the transcription of the protein is turned on by virus infection. And then it's activated by double-stranded RNA. And you may be saying, what is this double-stranded RNA coming from? Not all viruses are RNA viruses, right? Right. But double-stranded DNA viruses can transcribe both the top and the minus and the bottom strands, and that will make a double-stranded RNA. And that's what we think happens for many DNA viruses. And of course, many RNA viruses have double-stranded RNA as part of their replication intermediates. All right, so the PKR is induced and activated by virus infected, activated by double-stranded RNA. It will phosphorylate EIF2 alpha. You get inhibition of host translation. It's also phosphorylated EIF2 alpha is actually also an inducer of apoptosis, programmed cell death. So the cell will die from a combination of apoptosis and inhibition of translation. A good way to stop an infection. And this is so powerful that every virus has some way to inactivate the PKR pathway. Every viral genome that we've looked at has at least one protein to antagonize this. Otherwise, this is so powerful there wouldn't be any viruses around. So the bottom line here is viruses can get around anything. So let me give you an example of an antagonist, a viral antagonist. This is an RNA made by adenovirus called VaRNA1. It's a short RNA, and its structure is shown here, VaRNA1. And what it does is to bind PKR, but only one molecule of PKR is bound. So PKR cannot be activated because in order to get activation of PKR, two molecules need to bind to a double-stranded RNA. How brilliant is that, right? It's just amazing. So VaRNA is made abundantly in adenovirus-infected cells, and that prevents phosphorylation of EIF2 alpha. So no phosphorylation, and you get active protein synthesis, whereas in the absence of VaRNA, if the adenovirus-infected cell is making double-stranded RNA, if you didn't have VaRNA, you would get inhibition of protein synthesis. You could do this experiment. You can delete the VaRNA1 gene from the genome. You can make a virus, and that virus, it doesn't do very well because it can't inhibit cellular protein synthesis. So again, this PKR is induced by infection. It's turned on when the interferon system senses virus infection. It turns on, the protein is made, and then it's activated by double-stranded RNA. Here's a sampling, a smorgasbord, another schmor word, a smorgasbord of viral proteins that antagonize this process. And they antagonize at various steps. They counter inactivation of EIF2. Remember, EIF2 is inactivated by phosphorylation, and viruses need to have it active so that they can get translated. So someone asked, the PKR can still bind double-stranded RNA. It's just that via RNA1 binding, one of them inhibits mutual phosphorylation. Exactly. It inhibits mutual phosphorylation, right? Exactly. And so here are some viruses and proteins that inhibit this pathway. So EIF2 is the target, of course, of PKR phosphorylation. Vaccinia virus encodes a protein called K3L, which looks very much like EIF2 alpha. It basically distracts all of the PKR to phosphorylating it, and it ignores EIF2, and it gets around it. Herpes simplex virus encodes a phosphatase that takes the phosphates off as soon as they're put on. Now here's PERK. We've talked about PKR down here, right? The other kinase is PERK. And PERK is in the ER membrane. And normally it's not a dimer, it's a monomer. But when there's ER stress, right? And that's when you're making a lot of viral spike glycoproteins, for example. The cell does not like that. This protein senses that and it dimerizes. Here's the luminal part of the ER on top here. This will sense the viral proteins and it will dimerize. And then this part of the protein phosphorylates itself, becomes active, and now it can phosphorylate EIF2, just like PKR does. Anyway, there are viral inhibitors of PERK. And then there are a whole bunch of viral inhibitors of PKR. There are double-stranded RNA binding proteins that sop up double-stranded RNA, so it won't activate PKR. There are the antagonists of RNA that we talked about, like the VA RNA that will only bind one PKR. And then there are antagonists of the protein. So you see, very different viruses here are encoding counters of this system because it's so powerful. Viruses would not get anywhere if they couldn't counter this. So this is how this has evolved. Next question. PKR is an interferon-induced enzyme that is inactivated by what, leading to phosphorylation of what and inhibition of translation. And the choices are GDP EIF2 alpha, double-stranded RNA EIF2 alpha, double-stranded RNA EIF2B, single-stranded RNA EIF2 alpha, or none of the above. Most of you got double-stranded RNA EIF2 alpha. That's right. PKR is an interferon-induced enzyme that is activated by double-stranded RNA and leads to phosphorylation of EIF2 alpha. So it's not activated by GDP. It is activated by double-stranded RNA, but it doesn't phosphorylate EIF2B. It's not activated by single-stranded RNA. Double-stranded RNA EIF2 alpha. All right, our last foray into translation is about stress granules. You know, we all get stressed for various reasons, but cells get stressed. The ER gets stressed. If there's a virus reproducing, the cells get stressed, and they can sense it in a variety of ways. And one of the things they do is to build these things called stress granules. And you can see them in the cytoplasm of infected cells. They're very specific circular structures made up of a variety of proteins, cell proteins, including RAP55, G3BP, TIA1, TIAR. You would never need to know any of these, but you just need to know there are cell proteins that go to building a stress granule, and then the cell puts all the RNAs into these, and that shuts off translation. So here are some actively translating RNAs, and when the cell senses stress from a virus infection, builds stress granules and puts the RNAs in them. And they're quiescent, they're not translated. And it's not good for the virus, right? You have to get your mRNAs translated. So viruses encode antagonists of stress granules. So you can see these form early in virus-infected cells, and then they go away as viral proteins are made that antagonize them. And these are the viruses here that encode antagonists. You know, we got influenza, flavi, poliovirus, arenaviruses, alpha viruses, more flaviviruses. They're doing all sorts of different things, like The NS1 protein of influenza virus binds RAP55, which is needed to make a stress granule, so it blocks them from forming. Hepatitis C virus, as you'll see in a moment, it reproduces its RNAs on lipid droplets. And one of the components of this complex is G3BP. It needs it to make the reproduction site. So G3BP is essential for stress granule formation. So Hep C steals that away by viral proteins in red binding G3BP. Poliovirus actually cleaves G3BP. Cleaving means a protease cutting it, right? So now G3BP won't contribute to the formation. Let's see what else we have here. Here we have a variety of viruses. All these viruses here, their replication complexes require components of the stress granule. Here, G3BP. So alpha viruses and arenaviruses and flaviviruses, they bind up these components in different ways because they need them for their RNA synthesis. And at the same time, it pulls out the components so that you can reverse stress granule formation. So this is important because otherwise the cell mRNAs wouldn't be translated. And many viral genomes encode antagonists of stress granules. All right, enough translation. Let's do some metabolism. Let's do things that you thought you would never see again, like the Krebs cycle. I have to say that about 10 years ago, this appeared in Virology, and I thought I would never see it again after college. But it's actually quite relevant because to make a lot of virus particles, you need energy and you need stuff. What kind of stuff? Nucleotides, amino acids, fatty acids to build new virus particles, right? The cell has to keep making those. And you need energy. Single peptide bonds requires 4 ATP molecules. It's a lot of ATP. And some people say, there's plenty of ATP around. Well, there's some days I feel I have no ATP, so it's obviously regulated. And so what virus infection does is to impact metabolism to ensure that all of this stuff can be made. Let me just answer a question here. Someone said, do the viral mRNAs also get taken up into the stress granules? Yeah, that's the point. These are viral mRNAs in cell too. It doesn't distinguish to stop viral translation. Okay, here's the Krebs cycle. And remember, pyruvate comes in. Pyruvate's a product of glucose metabolism. We'll see that in a moment. You make acetyl-CoA, right? And that starts the cycle. You make all these intermediates. And along the way, you can make energy. And you can make intermediates for other things, like citrate goes off to make fatty acids. In fact, human cytomegalovirus infection pulls off the fatty acids from citrate because it needs it for its membrane. And that would kind of make the thing halt here if it didn't replenish it. So in human cytomegalovirus infected cells and also vaccinia virus infected cells and herpes simplex virus infected cells, they pull in glutamine, which they can convert to alpha-ketoglutarate and keep the cycle going. Because herpes simplex also pulls off from the oxaloacetate step, aspartate, which will eventually go to making pyrimidines and purines, right? Nucleic acid building blocks. So a number of viruses pull intermediates off, but they make sure they replenish them. In many cases, it's via glutamine here going in at that step. Otherwise, this cycle would halt, right? You pull out all the citrate, you're not going to go downstream. And so those are some of the impacts that virus infections have on metabolism. They need specific building blocks, and we'll look at some more of these in a moment. And they have to modulate these cycles, these biochemical cycles to do that. Many virus-infected cells show increased glycolysis, right? The breakdown of glucose. And that is shown in this experiment where we have plates of cells. And, cell medium has a pH indicator in it. So, when it's the right pH, it's this nice red or orange color. And then as the pH drops, it turns orange and then eventually yellow. And if, your cells are turning yellow and they shouldn't be, that's a problem. You can see it visually very easily. So here is a plate of cells and they're infected with adenovirus in the middle. And we're looking at different times after infection. And you see this virus is slow. It takes a long time to get going. But starting at about 48 hours, you see the medium is turning yellow. And that's because the low pH is a consequence of the production of lactic acid, which is a product of glycolysis. We'll see that in a moment. The mock-infected cells, what is a mock-infected cell? You just, instead of adding virus, you add PBS, buffer, and you go through the whole procedure and you put medium on and they're fine. And here is the cells are infected with an adenovirus mutant that cannot reproduce, the deletion in the E4 gene. And you can see it doesn't acidify the medium. And this is a different way of looking at it. This is actually glucose consumption. You can measure glucose consumption, nanomoles per million cells per hour. And you can see it goes up in adeno wild-type infected cells. So the cells are consuming glucose. We sort of had evidence for that here in the low pH, but here you can see it quantified. And the E4 deletion mutant is not consuming any glucose. Yeah, it's like a negative control to make sure you didn't do anything that would make the cells turn yellow. Who knows? You know, you could have dropped something in the cells or whatever. You always have to have this negative control. And sometimes you'd like a positive control, but we don't. In this case, we're looking at what the virus is doing. So many other viruses are increasing glycolysis, rhinoviruses. I wrote an article once called Rhinoviruses Have a Sweet Tooth. They like glucose. If you take it away, it inhibits reproduction. So here is glucose metabolism. Glucose, of course, is the major breakdown product of what you eat, the carbohydrate in your food, and it's imported into cells. So here's glucose at the top. This is the plasma membrane of a cell. And there's a transporter called GLUT34 that pumps the glucose into the cell, and then it goes through this metabolic pathway. And, at different points, you make energy. It's eventually converted to pyruvate and gives you 2 ATP molecules and 2 NADH. And some of these intermediates that peel off, you can get more ATP made as well. And virus infections certainly perturb this, right? Some of them increase it. Some of them don't do anything to it. But I just want to point out that, as you go through these intermediates, you can pull off here at the glucose 6-phosphate point, you can pull this off into what we call the pentose phosphate pathway. And ribose 5-phosphate is a precursor for nucleotides. So this can give rise to nucleotides. And then down here, acetyl-CoA, which can be made from pyruvate, is a precursor for fatty acid synthesis, which of course you need to make membranes, and many envelope viruses need that. In fact, many non-envelope viruses need membranes. They need the membranes to reproduce their RNAs on. And finally, the last step gives you lactic acid, and that reduces the pH of the medium. So the adenovirus is somehow pushing glucose through this shunt at a higher rate than uninfected cells, because the uninfected cells are just, the medium is still yellow, is still red. So let's take a look at one example of perturbation. Here are 2 herpes viruses, herpes simplex 1 and human cytomegalovirus. And they have different effects on glycolysis. Herpes simplex virus doesn't really increase the uptake of glucose. So this green arrow here means human cytomegalovirus is increasing the uptake of glucose, and it pushes it all the way to acetyl-CoA, which it needs for membranes. So the idea is that HCMV is kind of a slow replicating virus. It's pushing all of the glucose to precursors of fatty acids, whereas in herpes simplex infected cells, the virus doesn't increase glucose uptake, but rather takes everything that normally would come into the cell and shunts it off to the ribose 5-phosphate pathway. It doesn't go on to pyruvate, but it wants to make a lot of nucleotides. And the idea here is that herpes has a shorter reproduction cycle. It needs more nucleotide precursors to make a lot of DNA, and it can get along with the pools of lipids that are present in the cell. If the viruses pull out the products of the Krebs cycle, how come lactate is elevated? Well, lactate is elevated because they're pushing them through here, and then the pyruvate has to go into the Krebs cycle in order to make more energy and so forth. But if you go to lactate, you will acidify the pH. Now, let's take a look at fatty acid synthesis. The problem here is that acetyl-CoA, which is made in the mitochondrion here, right, there's your mitochondrial membrane. So the glucose goes to pyruvate, which is imported into the mitochondria, is converted to acetyl-CoA. It can't get back out because in the cytosol is where fatty acids are synthesized. So how does it get out? Well, you do a little trickery here. You convert it to citrate, which can then get transported out of the mitochondrion, And then in the cytosol, you convert it back to acetyl-CoA, which costs energy, but that's what you have to do. And then it can get converted back to pyruvate and go back in if need be, or it can go off to this pathway, malonyl-CoA, and then fatty acid synthase will make fatty acids from this. Okay, so there are a number of enzymes here. And in fact, in human cytomegalovirus infected cells, most of the citrate leaves the mitochondria for fatty acid synthesis, because as we saw in the previous slide, this virus requires a lot of membranes. So it pulls out the citrate and makes fatty acids with it. And if you inhibit malonyl-CoA synthase or fatty acid synthase, and then you infect cells with cytomegalovirus, you inhibit virus yields. So that provides direct support that these fatty acids are required for production of virus. And for another virus, vesicular stomatitis virus, another member of this shuttle is increased in cells infected with that, again, because this virus requires fatty acids. A little more on alterations here. How about viruses that infect the liver? Could they change glucose metabolism and cause disease? So here, of course, the liver is where you make glycogen and where you make glucose. Gluconeogenesis happens in the liver, right? Now, hepatitis B virus is a liver-specific virus. And infection with that virus is associated with development of type 2 diabetes, which, insulin deficiency resistance type diabetes. And that is in part because infection stimulates levels of enzymes involved in glucose synthesis. It requires glucose for, presumably energy and also fatty acid precursors. So increases the production of glucose, and that leads to diabetes. Hepatitis C virus does a similar thing. Now, Hep C reduces uptake of glucose, so to make up, it increases gluconeogenesis in the liver, so you get high levels of glucose, and that leads to diabetes. So these changes induced by virus infection have consequences for pathogenesis of human diseases. Back to our citric acid cycle, which of course is the hub of carbon metabolism. And as I already mentioned, it makes precursors for many things like fatty acids, precursors to nucleotides and so forth, and energy, of course. And virus infections impact this. One example is lipid metabolism. Now, here's a lipid, a triacylglycerol. These are the primary energy source in cells. They can be oxidized to give rise to energy, and it's how we store energy in these triacylglycerols. And of course, viruses require them for membrane synthesis. And so lipid metabolism is modulated in virus-infected cells. You have to have a balance between oxidation and synthesis, whether you need the energy, which would be provided by oxidation of lipids, or if you need them synthesized for membrane. So you have to have a balance. And just to show you what happens here, these triacylglycerols, they're in your bloodstream. They bind lipoproteins, which in turn bind receptors on cell surface, a low-density lipoprotein receptor. Lipoprotein lyase, it's the wrong name, lipoprotein lyase, which breaks up lipoproteins into free fatty acids, and they get imported into the cell, and then from them are made acetyl-CoA synthase, makes acetyl-CoA, which can then be imported into storage areas, including these so-called lipid droplets. These are collections of acetyl-CoA, and they're coated with a protein. So these are called mature lipid droplets. And it's a way to store fatty acids in the cell. Human cytomegalovirus infection induces the synthesis of very long-chain fatty acids for assembly. The fatty acids come in different lengths according to the chain, short, medium, and long. So this virus likes long chain. And what it does is increases carbon flux from glucose to acetyl-CoA, which we've kind of mentioned already before. So you get increased fatty acid production, but also the long-chain fatty acids that we find in viral membranes. And the mechanism involves increased activators for transcription of genes that are needed for lipid synthesis. And that's shown on this slide here. Let me go through this with you. So the genes for fatty acid and sterol synthesis, of course, they're in the nucleus. Their transcription is regulated by this protein, SREBP1-M. So this is a transcriptional activator that will turn on the synthesis of these genes, and then you can make fatty acids. Now, normally, this SREBP1-M is stuck in the ER here. There it is right there. And it is it's attached to this double membrane, transmembrane protein here, and then in turn it's bound to a protein called SCAP, and it stays there. It doesn't move, so it can't get in the nucleus. Now, in human cytomegalovirus-infected cells, that virus, of course, makes glycoproteins in the ER. It causes ER stress, which causes the PERK to dimerize. That's the ER kinase. And when PERK dimerizes, it pulls this protein off of SCAP SREBP1, and that allows it to then move through the Golgi. And finally, at the last part of the Golgi here, this protein is cleaved off, and you have SREBP1M going into the nucleus and turning on genes for fatty acid synthesis. So this is turned on. Normally, this would be regulated by cholesterol or fatty acid levels. You know, if you have plenty of fatty acids around, this protein would remain in the ER. But virus infection causes it to come out and go in the nucleus, turn on genes for fatty acid and sterol synthesis because the virus requires those, of course, for reproduction. Now, hepatitis C does a similar thing, and what it does is to balance oxidation versus lipogenesis, right? So lipids can be made or they can be degraded to make energy, and two different transcription factors control the genes for synthesis or oxidation. And so Hep C decreases the amount of transcription factor FOXA2 that's needed for the genes that cause oxidation. It reduces oxidation, and it turns up FOXO1, which is needed for transcription of genes that increase lipogenesis. So the virus does this. And the consequence is that in virus-infected cells, you see more fatty acids. So here are hepatocytes. On the left, mock-infected hepatocytes. They're stained with DAPI for the nucleus, and then there's a stain for a fatty acid, which shows up green. So you can see there's a little bit of fatty acids in these cells. And then if you infect them with hepatitis C virus, the amount of fatty acids goes up. And again, that's because the virus is controlling these transcription factors to favor lipogenesis and inhibit fatty acid oxidation. Our last question for today is, how might virus infection lead to increased levels of ATP? Stimulation of glucose uptake, increased glycolysis, increased oxidation of fatty acids, increased utilization of glutamine, or all of the above? All right, we're stuck. So let's see what we did. 38. And most of you got all of the above. That's correct. They're all right. You can get increased levels of ATP by stimulating glucose uptake. We saw an example of that, increased glycolysis, increased oxidation, increased utilization of glutamine, which goes into the TCA and the Krebs cycle and replaces if you take out citrate. Now, when many viruses infect cells, especially RNA viruses, they remodel the cellular organelles. And here's an example in poliovirus. And all these viruses make some kind of membranous structure on which to reproduce their RNA genomes. And here is, again, that stain for fatty acids in mock and poliovirus infected cells. And you can see increased import of fatty acids into polio-infected cells, increased synthesis. And what happens is you get the formation of these double membrane structures, and on the surface and inside of those, the viral RNA is reproduced. And this is done by a few viral proteins that grab lipid droplets and shunt the triglycerides into these membrane components. So it's an active process by a couple of viral proteins. So again, on the surface of these vesicles, the viral RNA synthesis occurs on the surface and inside. And late in infection, these can trap virus particles, and they can get released by non-lytic mechanisms, as we discussed. A couple more examples of this, because different families of viruses do this differently. Here's hepatitis C virus. They're really interesting because these assemble on lipid droplets, those storage areas for lipids. And early on in evaginations of the ER, the viral genome is reproduced. Here's some viral RNAs, double-stranded RNAs being made. And then later in infection, a lipid droplet comes in here. It's coated with viral proteins, and you can see a viral protein, a virus capsid being made by budding into this lumen up here, and it's grabbing a viral RNA. And you can see these very clearly in infected cells. Here's a reconstruction of an electron micrograph in different colors, and you can see these extensive vesicles linked to the endoplasmic reticulum. And again, these kinds of structures are induced by viral proteins as sites of viral replication and eventually assembly of particles. The last one I want to tell you about is so cool. It's a plant virus, which It's an RNA virus. It induces these vesicle evaginations from the ER, and viral RNA synthesis occurs in these vesicles. You can see it happening right here. The viral polymerase is in there. But these vesicles are coated with a viral protein, p33, which grabs pyruvate kinase and pulls it into these vesicles and uses it to make ATP. So pyruvate kinase takes phosphoenolpyruvate, makes pyruvate from it. You get a molecule of ATP from that. And of course, the polymerase needs energy to make RNA, and that's where the energy is coming from. The virus pulls this enzyme into these vesicles by virtue of this viral protein p33. And you can see that in this fluorescence micrograph series here. This is an antibody against, these are infected plant cells. The virus is tomato bushy stunt virus. And here we're staining with antibodies to this p33 protein. And here is pyruvate kinase tagged with green fluorescent protein. And the merge shows you, and there's the blow up, one of these vesicles where you've got p33 in there along with pyruvate kinase. And so again, the viral RNA synthesis happens and it needs energy, and the pyruvate kinase provides that. Amazing things happen in virus-infected cells. And of course, we can't end this without referring to coronaviruses, which also reproduce their genomes on double membrane vesicles that are induced in infected cells by viral proteins. Here's a electron micrograph reconstruction of them. You can see many of them made here, again, made in infected cells. And on these are made viral mRNAs, and the genomes are replicated. So in cells infected with most RNA viruses, you see these alterations. The exception would be, of course, reoviruses, where the RNA synthesis occurs in the particles, so they don't make these double membrane vesicles. So we have actually finished the reproduction cycle of viruses. That's the first half of this course. And the rest, we're going to talk about diseases and how to prevent them. So first, we will go through some infection basics. Don't miss it. You want to learn all about transmission and shedding and so forth. Come back for that one.